Good morning and good evening, everyone. So my name is Iwona Hawryluk, and I'm a data scientist in Chrome Global. And I'd like to welcome everyone to a Data Science for Food Safety webinar. So I'll start with a brief overview of the content of this webinar. So first, I'll say a little bit about Chrome Global, about what we do and who we are. And then I'll talk about how we can use data science in food safety. And I'll discuss some common data traps and how we can solve these problems. Then I'll explain what is probabilistic modeling and how does it differ from different approaches typically used. I'll present some case studies in microbial food safety and packaging. And finally, I'll talk about the total aggregate exposure model. So about Chrome Global, we are a data science technology company, and we help companies make better decisions using data, and we make data science easier to grasp for both scientists and non-scientists. We have expertise in food science, including food safety and nutrition. Also, we have expertise in cosmetics ingredients exposure and cloud computing. Our services include technical services such as data analysis and writing scientific reports. We also develop predictive models that can be deployed on our cloud-based platform called Expert Models, which enables the users to run the analysis themselves. We also have off-shelf software data science tools that we can license or customize. We provide data management and data gathering services as well. So firstly, I would like to answer the question, how can we use data science in food safety and what can we achieve with that? So using data and predictive modeling allows us to predict the risks that consumers are facing in various areas of food safety. And the three areas of food safety that I will talk about today are chemical, microbial, and packaging safety. Chemical safety concerns the consumer's exposure to the various compounds that are hidden in the foods that we eat every day, such as some additives like sweeteners or flavors, but also contaminants and pesticides residues. Microbial safety refers to exposure to microbial contaminants, but it also involves predicting the pathogen evolution in the food and in production facilities. We can also use microbial data to predict the shelf life of the products. Finally, packaging safety focuses on the contamination of the foods with the compounds that migrate from the packaging into the food. What problems can we solve using predictive modeling? So we can use it to inform product development. For new substances and new packaging types, we can use it to make sure that the new product is safe for the customers. We can also assess the current risks and consumer safety in the population. We can run and compare different scenarios, for example, to see what is the optimal concentration of the ingredient in the product, or to identify subpopulations that are at most risk. We can also use historical data to predict the outcomes instead of running tedious lab experiments. And finally, we can assess the impact of a product or process change on the consumers. So now I would like to talk a bit about data limitations and solutions. So data that we are working with has various limitations that we have to be aware of to ensure that our predictions are as accurate as possible. The most common problems we have with data is the quality of the data. So if the data is of low quality, then the model that's using this data will make poor predictions. And one of these issues that we have to deal with almost every time we get the new data set is messy data. And that means that the data is missing a lot of values or has mixed data types. And this is often the case when we're working with the industry data, because it's usually collected by many people and each of them might have different habits when it comes to collecting the data, and also the structure and framework of the data collection might not be clearly stated. Another quality issue with data is dealing with unstructured data, such as data that's in books and images, and it's surrounded by a lot of information that is irrelevant to us. So solutions to these problems are various data processing techniques. So a lot of work of data scientists is spent on data cleaning and structuring. There's also various new data mining techniques, such as text mining or OCR, optical character recognition. These are machine learning methods that can help us to go through large volumes of unstructured data, and they can quickly extract the information that we are looking for. Another type of data limitations are data gaps. So a common problem in the food safety assessment is lack of occurrence data. So this happens when we have the distribution of the concentration of the chemical, but we don't know whether this chemical is present in every single product. Another limitation is data that's not representative. For example, if we have brand-specific data, it might not be representative of the whole market. And finally, there's a lack of surveys that would report on different types of habits, such as both food consumption and cosmetic usage. Some ways to fill these gaps are using alternative data sources, such as regulatory data or market research data. We can also use custom data gathering tools, such as developed in Creme Global Foodbook 24 or Creme Diet. We can also combine different data sets. However, this has to be done with caution because, for example, it's not wise to mix the behavior data of American and European consumers without applying any adjustments. The last type of data limitation that I will talk about is the complexity of the data and the models. So one example is that people's consumption habits are very complex, and they are very closely related to the age and the country of the subject. So we need a lot of country-specific data to run assessments. Sometimes we might have data of good quality, but the size of the data set might be too small for our results to be statistically significant. There might be also multiple routes of exposure to one chemical, so sometimes one model is not enough to understand all of the risks in the population. There's also another problem is lack of the knowledge of the field. And it's really difficult to sense check the data or results if we don't have the full understanding of what they mean. As a solution, we can apply different statistical methods. For example, we can extrapolate and interpolate from available data. So if we know a couple of values, we can create distributions to cover the whole range of possibilities. As a solution for a small data size problem, we can apply Monte Carlo simulations, which is one of the features of all the models developed in Creme Global. We can also use bespoke models and approximations depending on our problem, and we can leverage multiple models if we have many routes of exposure. So next, I will talk about the types of data sources typically used in the food safety assessments. This slide shows the structure of the data in the food consumption surveys. So firstly, we need the subject-specific data. It contains basic information about every subject in the survey, and normally it consists of more than 1,000 participants from one country. For each of the subjects, we need their consumption diary, which is a record of all individual foods they consumed in a day, together with the amounts consumed. Then these individual foods can be grouped into food groups depending on our needs and depending on the level of granularity desired. Finally, for each food group, we need the ingredient or contaminant concentration. And similar methodology works for assessing nutritional intakes, but in that case, we use nutrients amounts instead of chemical concentrations. So now let's take a closer look at the concentration data. The concentration of the chemicals of interest in foods can come in various forms and can come from many sources. So such forms of data are typically either single point estimates of the concentrations, or these values can come from known ranges or empirical and parametrical distributions. Most common sources of data is industry data, but it can also be found in the publications and reports. However, if no distribution of values is available, then often we need to use maximum values allowed by the regulatory bodies, just to make sure that we are not underestimating the risks. Now, ways we can segment the population. So consumption surveys also carry detailed information on the subjects that took part in the study. So usually this information covers basic demographics, such as gender and age, but it often comes with additional data, such as genotype or income status. And this data is crucial to understand the risks concerning the food consumption in all the subpopulations. Because, for example, it is not reasonable to put children and adults into one category, as they consume very different foods and in different amounts. Okay, so now that we know about the underlying data, I will explain what is probabilistic modeling and how it can be used for chemical food safety. To understand the dietary risks in the population, we carry out exposure assessments. And this way we can find out how much of this chemical that's hidden in the foods people consume every day. And we achieve it by combining the information about food consumption and concentration of the compound. And in this way, we can see the exposure levels in the whole population. This methodology is applicable across all kinds of chemicals and additives, but also nutrients. When we have the distribution of exposure levels in the population, we can look at the consumers who are highly exposed and compare it to the reference dose. So usually we look at the 95th percentile of the exposure. Depending on the data that we have available and our approach, we can go from very crude worst-case scenarios down to refined probabilistic exposure modeling. So to obtain the worst-case scenario, we often use deterministic model, which is using maximum values of concentrations and amounts. The probabilistic method, on the other hand, takes into account various consumer habits and practices, and also it uses whole distribution of data, giving us much more refined assessments and reflecting the reality much more accurately. So in the deterministic model, the worst-case exposure levels from each category, and typically we look at the 95th percentile, they're stacked on top of each other to give us the total exposure. So that means that to calculate the aggregate exposure, we sum up the highest exposure levels from each subcategory. However, we have to remember that the high consumers in each of the subcategories are in fact very different consumers. So, for example, a person that eats a lot of apples might be eating very small amounts of onions. And this is taken into account in the probabilistic method, where aggregation happens on the subject level. So high consumers in each category are really indeed different people. Another difference between the deterministic and probabilistic approach is the type of data used. In the probabilistic model, the aggregate exposure is based on the actual product consumption and full distribution of data. So in other words, in the deterministic case, the point estimates are used to give very conservative and worst-case exposure levels. Whereas in the probabilistic case, we can make use of the whole distribution of data to make the assessment much more refined and better reflecting the reality. And this approach can be used for the amounts of products consumed as well as the concentrations of the ingredient or contaminant in foods. So now I will show how the probabilistic exposure model actually works with food consumption surveys. And so for each subject, we have a detailed record of consumed foods that is obtained from the survey. And this contains information on the specific type and amount of the product eaten. To each of these consumption events in the model randomly assigns a concentration of the compound. It can be either done by, a point estimate, single value, or it can be done by sampling from a distribution of concentration. Then we can multiply the amount by the concentration to get the amount of this contaminant or chemical that's eaten in each of the consumption events. And this is done separately for each day and each subject in the consumption survey, which is very important because every person's eating habits are different. Then the daily average exposure is calculated individually for each subject. Finally, we can rank the subjects from the least to the most exposed consumers to get the full picture of the exposure levels in the population. Depending on our needs, we can look at the average or the median consumer exposure, or we can look at the high or 95th percentile consumer exposure level. To visualize what I just talked about a bit more, you can see here the hypothetical example of using this worst case deterministic approach versus refined probabilistic approach. Just to give you an idea of how these two approaches can compare. So typically the deterministic approach, shown here with the blue line, gives higher exposure levels, and they can exceed the health-based thresholds such as acceptable daily intake. With the probabilistic approach, on the other hand, that is represented here with the orange line, we get much more refined and realistic outcomes because we're using the whole distributions of data and we are aggregating on the subject, not product level. The model I just described is already implemented in our cloud-based platform called CREM Food Safety, which allows the users to easily run food safety assessments. So CREM Food Safety is ready-to-use software with a lot of data already built in. The data sets are chosen as inputs and then combined by the model to allow detailed analysis for various demographic groups, and it provides detailed food risk assessment. CREM Food Safety is based on validated scientific data, which is used and accepted by regulators internationally. Also, the tool can be easily customized and supports collaboration between the members of the team. It also has capacity to handle large amounts of data. So as I mentioned, a lot of data sets are already built in CREM Food Safety. Such data sets are the consumption surveys from the countries shown in blue on this map. Some consumption surveys are available as well, but they require additional permissions. However, if you are interested in the dietary habits in different countries, there are some ways of getting some more data. So such sources of alternative data can be, for example, market research data, from which we can get the market shares or occurrence data. We also have a lot of crowdsourced platforms with a lot of interesting data sets. And we can also use online gathering tools such as developed in CREM Global Foodbook 24 or CREM Diet, which allow to gather consumption data in the populations. So next I will show the two case studies of our work in the field of microbial food safety. Microbial Stability Calculator is a software developed for one of our clients for predicting microbial stability of carbonated and non-carbonated beverages. It takes as an input a number of parameters such as pH, carbon dioxide volume, and acid concentration. Using this data from the past and statistical methods such as logistic regression, the tool calculates the probability that the product will remain stable. So this microbial stability calculator can be used for modeling shelf life using the real data instead of long and tedious lab studies. The SAFE project, which stands for Sequencing Alliance for Food Environments, is a result of partnership between the University College Dublin Centre for Food Safety, Creme Global, and 6 leading food and nutrition companies. The aim of the SAFE project is to build a predictive model using DNA sequencing data that would help us to understand the microflora on the production plant. Specifically, the focus of the SAFE project is on facilities producing dairy products and packaged meat. The idea behind the SAFE is that particular species of bacteria can make the environment favorable for other species to grow. And we would like to know whether detecting one type of bacteria could predict the presence of other species. The ultimate goal of this project is to reduce the risk of bacterial contamination in the food supply chain. Now I would like to talk about the packaging safety case study. So the study I'll talk about is called FACET, which stands for Flavors, Additives and Food Contact Materials Exposure Tool. This is a European Union framework project that Creme Global participated in collaboration with industry, government and academia. The goal of FACET was to develop a publicly available software for calculating the exposure to chemicals in the packaging materials, including additives and flavorings across 8 European countries. The system consisted of a number of unique databases and models. And one of the components of the software was migration model that I will describe in a bit more details. So the safety issue that the migration model is addressing is the fact that particular compounds of the packaging materials can migrate from food packaging directly into the food. And this poses a risk that some chemicals that people should not be eating are consumed accidentally. There is a number of properties that can influence how the compound migrates from packaging to food. And these properties characterize the type of packaging, the type of food, and also the storage conditions. And all these things have impact of how much of the unwanted compound is transferred from the packaging into our food. Using the migration model, we can simulate the migration of these chemicals in each layer of the packaging and into the food, as shown in this figure. We can use historical data and predictive modeling instead of running lab studies again. In the last part of this webinar, I will talk about the total aggregate exposure model. So what exactly is total aggregate exposure? So during this webinar, I talked about the exposure to the chemicals that are present in the foods and consequently consumed by people. However, food is often not the only source of the exposure. There are some compounds that are present not only in the food, but also in cosmetics and personal care products, or even tap water. A good example of this is the exposure to pesticides residues. So we are exposed to pesticides residues when we eat the food, but we are also at risk of inhaling or getting in contact with them when we spray the plants in the garden, or we pet a dog which is wearing an anti-tick collar. If we want to get a realistic and a comprehensive overview of all the risks related to a particular chemical, we have to look at the broad picture and consider all the routes of exposure. An example of applying total aggregate exposure is a project that Creme Global did recently for a company called Microbite, which is a company specializing in microbial control products. So Microbite asked us to quantify the exposure to the pesticides residues in the US population in 5 food crops and tobacco. So the pesticides residues sprayed on tobacco crops might be inhaled by the smoker when they smoke cigarettes. So we wanted to estimate what is the cumulative exposure to these pesticides that are both consumed and smoked. This was quite a complex project which required employing various data sources. So we used NHANES, or National Health and Nutrition Examination Survey, that provided us with dietary habits of the US population. Enhanced also contained information about smoking habits of each of the participants of the survey. Now, the pesticides concentration data was obtained from the PDP, or Pesticides Data Program, which is a program that monitors the levels of the pesticides residues in the US that are found on everyday foods. For the levels of pesticides residues on tobacco leaves, we estimated this with guidance residue levels provided by Coresta. So, because in this scenario the exposure was due to both ingestion and inhalation, we had to integrate two different models in this analysis. We used Kersen-G dietary model, that is a model developed in conjunction with a wider chemical industry, including Environmental Protection Agency. So Kersen-G allows to calculate dietary exposure to the pesticides residues in food commodities in both chronic and acute timeframes. It uses probabilistic calculations to give an aggregate and cumulative exposure, that is exposure to either one or multiple chemicals. For the inhalation exposure, we use the two-box model that I will show on the next slide. So this presents the schema of the two-box model. Two-box model was primarily developed for assessing exposure to ingredients of sprayed cosmetics, such as perfumes or hairsprays. We use the same model because also in the case of smoking a cigarette, there is one single source of the chemical and exposure is due to inhalation. So I'm not going to go into details of how the two-box model works, but I just wanted to show you how we can incorporate multiple models to accommodate for different exposure routes. So as I explained before, we use the consumption diaries of the enhanced participants to assess the dietary risks. And the amounts of food eaten were multiplied by the concentrations of the pesticides residue data for each of the consumption events. Then for each subject that was a smoker, we calculated the exposure to pesticides residues using the two-box model. The results of the both assessments were then combined on a subject level, which gave us a very refined analysis because we didn't have to make any assumptions on the average number of cigarettes smoked, because that came directly from the recorded data. That's all I wanted to talk about today. So thank you very much for listening, and I hope you enjoyed it. I hope you got some new information about using data science in food safety. And if you had any questions or would like to talk to us about Creme Global's data science services for food safety or possible collaborations, or if you'd like to get a product demo of the Creme food safety software, please drop us a line to one of the email addresses given on this slide. Thanks again and goodbye.